Assalamualaikum and hi to all. So, first, let me introduce myself. Okay, my name is Nuri Nazia Muhammad Noor, and I will introduce about paper chromatography of pigments in spinach leaf. So, basically, chromatography is an analytical method permitting the separations of a mixture into its molecular components. In this technique, a concentrated spot of the pigment mixture is deposited at one end of a paper strip. The paper strip is called the stationary phase. The ends of the paper strip containing the pigment mixture spot is then placed in a beaker containing a small amount of solvent, which is called the mobile phase.
The first theory of the technique used is Spinach chromatography. Spinach can be used to demonstrate how different compounds within a plant extract can be separated based on their chemical properties. We can determine this within three methods, which is extraction, RF values, and separation by polarity. Spinach are referred to the plant materials as a source of extracting pigments, especially chlorophyll. The stationary phase in this method is paper, and the mixture of the spinach leaf act as a mobile phase. The purpose of this method are to separate the mixture based on differential pigments travels at different rates, causing to the separation into distinct bands. The second theory of the technique used, which is TLC technique. TLC stands for thin-layer chromatography. It is that compound in a mixture separated based on their relative affinity for the stationary and mobile phase. This method used in this technique is visualization and separation. This technique used to identify compounds in a mixture, determine the purity, monitor reaction progress, verify small amounts of compounds, and lastly to analyze many different types of substances. In this method, there is a few tips for you. To avoid streaking, add a few percent of acetic acid or formic acid to the solvent, or a few percent of triethylamine for bases. And to remove solvent, use vacuum chamber for non-volatile solvent. In my left, there is an example of TLC. You can see the mobile phase, TLC plate used that placed in the TLC chamber, and the spotting line in the mobile phase.
Objective for the experiment is to identify the spinach present by using paper chromatography and to separate compounds in mixture by using thin layer chromatography TLC technique.
Hi and Assalamualaikum. My name is Muhammad Firasul bin Zulkifli. So today I would like to introduce the material that used for chromatography experiment, and we need the chromatography paper, TLC paper, beaker 500 ml, filter paper, mortar and pestles, spinach, retort stand, measuring cylinder, sodium chloride, hot plate, Erlenmeyer flask, acetone, isopropanol, calcium carbonate, anhydrous, and 50 ml beakers.
For this slide, I will explain the method to conduct a chromatography experiment using the spinach technique. This method allows us to separate and identify pigments present in spinach leaves. The first step is to prepare and tear a few fresh spinach leaves. This ensures that we have enough sample material for the chromatography process. Next, we cut a strip of chromatography paper and draw a 2 cm baseline near the bottom of the strip. This will serve as the reference point for our sample application. Using a dropper, we place a small drop of the spinach extract onto the baseline of the chromatography paper. It's important to let the sample dry for about 5 to 6 minutes to ensure proper absorption. Pour 70 ml of NaCl solution into a beaker until it covers the marked line inside the beaker. The solution acts as the mobile phase that will carry the pigments through the paper. Insert the chromatography paper into the beaker and clamp it to a rated stand, ensuring the paper hangs freely without touching to the sides. Allow the solvent to rise up the paper until it reaches halfway. Method to conduct the LC paper. First, take a few spinach leaves, remove any stem and chop into small pieces. Place the chopped leaves into a mortar. Add acetone and grind the mixture thoroughly. Next, filter the mixture through filter paper to collect the pigment extract into a small container. Take a TLC plate and draw a baseline approximate 1 cm from the bottom edge using a pencil. Using a capillary tube, apply a small drop of the spinach extract on the baseline. Let it dry and repeat the concentrate the pigment. Pour acetone into a chamber until the depth is about 0.5 to 1 cm. Place filter paper inside the chamber so it touches the solvent and lines the walls. This will saturate the chamber with paper. Cover the chamber with a lid or watch glass and let it sit for a few minutes to equilibrate. Carefully place the TLC plate upright in the chamber, ensuring the baseline stays above the solvent level. Once the solvent levels up the plate, remove it and let it dry in well-ventilated area. Observe the separated pigment bands on the plate. These bands represent different pigments from the spinach leaves. Lastly, calculate the RF values.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Hazim Razali and I'm going to explain about the explanation of applications for paper chromatography. First is plant pigment analysis. It used to separate and identify pigment in spinach leaf, and the result will be come out. We can identify the chlorophyll A, chlorophyll B, carotenoids and xanthophytes, and it can be useful for understanding the photosynthetic process and the role of pigment in capturing light energy. Second is for educational demonstrations. We usually use this in the labs to teach the student how to use it. We commonly use it in school and college labs to demonstrate basic chromatography principle. It also helps students visualize the separation of compounds based on solubility and affinity. And third is preliminary research. It's very quick and inexpensive method to analyze plant pigment composition before conducting advanced analysis. It's also very useful for screening plant health and the impact of environmental stress on pigment profiles. So lastly is for very environmental studies. Identifies pigment degradation due to pollution, UV exposure or other environmental factors, which is we can use to detect in our environmental by using this paper chromatography technique. And now I'm going to explain about the application for thin layer chromatography, which is TLC. And first is pharmaceutical testing, which is to identify plant-based bioactive compounds which can be isolated for medicinal purpose. Screen plant extracts for natural antioxidant, flavonoids and alkaloids. That is for the example. And second is food quality control, which is we can find in many factory for the produce food, which is they use to detect artificial colors, natural pigments and pesticide residues in food products derived from plants. And third is forensic science. They use to identify trace plants in pigments or dyes in forensic samples. It's very useful for analyzing botanical evidence at crime scenes. And lastly is for it can be used to chemical purity testing, which is to determine the purity of compounds extracted from plants, ensuring quality in industrial process.
So this is the advantage of spinach techniques. First, spinach techniques is high chlorophyll content and it makes it ideal for studying. Second, spinach extracts can replace synthetic reagents in certain organic reactions. And the third one is spinach techniques is ideal for learning techniques like extraction, purification and characterization of organic compounds. So, we will talk about the advantage of TLC techniques. First of all, TLC is a quick method that can analyze many samples in a short time. TLC can easily separate and recover components from complex mixtures, and TLC can be used for a variety of purposes, such as screening tests, pharmaceutical production, and environmental toxicology. So everything must have the pros and cons. So we're gonna dive into the disadvantage of spinach technique. Spinach techniques, for the main source is natural product that contain a complex mixture of compounds, which can make it difficult to isolate a pure product due to its complexity. And the chlorophyll or carotenoids are natural compounds that sensitive to light, heat, and also oxygen. Improper handling can affect the results. The desired compounds are often present in small quantities, requiring large amounts of spinach or the consuming extraction processes to obtain a sufficient amount for analysis reaction. We're gonna get into the disadvantage of TLC technique. TLC can be inconsistent in detection, and TLC can also perform qualitative analysis, not quantitative analysis. If the solvent level is too high, the compound will dissolve in the solvent instead of migrating up the plate. So this is the disadvantage of TLC technique.
Chromatography is a strong and easy way to see and examine the makeup of complicated combination, forensic analysis, quality control, and scientific research all make extensive of this method.